Dear viewers, my name is Alexander Voss, CEO of VectorY. We would like to thank the organizer of the Cell and Gene meeting on the Mesa for the opportunity to present VectorY here today. VectorY aims to develop innovative therapies to treat neurodegenerative and neuromuscular disorders that have a high and growing prevalence worldwide. We aim to do so by attacking the proteinopathies that lead to neurotoxic protein misfolding and aggregation and subsequent clinical presentation. VectorY combines the therapeutic efficacy of antibody technologies with AAV-based gene delivery, using vectorized antibodies, short-chain fragments, and structurally engineered AAV capsids and transgenes with improved target and tissue specificity. We use proprietary AAV manufacturing processes and also a proprietary dedicated GMP infrastructure. We have already established a strong foundation for future business growth by assembling a very strong management team and a strong capital foundation with Forbion and the closing of a 31 million euro seed round in June of this year. As you know, there's a high prevalence and high unmet medical need for neurodegenerative diseases in the major markets. Over 25 million people suffer from progressive Alzheimer's and Parkinson's in 8 major markets alone. Also, orphan diseases like ALS and DM1 alone each have about 60 to 100,000 patient population to treat. These diseases have a growing incidence of about 0.5 to 1% per year, largely related to aging of the population. For all of these diseases, a high unmet medical need persists, and VectorY focuses on ALS and other orphan diseases as an initial proof of concept, but we believe that there's also application for our technologies in the broader disease areas. The neurodegenerative diseases are commonly caused by protein toxicity. Some examples here: Alzheimer's disease, Parkinson's disease, Huntington's disease, and ALS. Each one have known protein pathologies associated with them, like beta amyloid, tau, alpha-synuclein, Huntington, and TDP-43. There can be a direct genetic link for each one of them, but also proteinopathies will be associated in a multiple fashion across these diseases, so not a single protein will play its role in each single disease. Several mechanisms underlie protein toxicity in neurodegenerative disease. In this simplified cartoon in the middle of a neuron, you see in the brown bulbs the aggregate formation. So these are protein aggregates coming into the cytoplasm that tend to clog together. They assemble and go to the mitochondria, to the vesicles, and also lead to impaired axonal transport. And also the synaptic function can be severely deteriorating. These effects can be due to external factors like aging, infection, inflammation, external stress, or toxic exposure, and lead to transcriptional dysfunction, and then subsequent protein aggregation can be leading to significant oxidative stress in the cells and leading to mitochondrial dysfunction and ultimately cell death. Also, the ability for the cell to maintain proteostasis in the stress granules can be impaired, and as a result, aggregate formation can be accelerated. And finally, due to the aggregate formation, the usually protective glial cells can recognize these aggregates as foreign and therefore start to exert a toxic effect rather than a protective effect. So all of these efforts accumulate and lead to further neurotoxicity. One important example here is amyotrophic lateral sclerosis, ALS, a well-known disease and the most common adult onset motor neuron degenerative disorder known. It's characterized by having the voluntary muscles no longer receiving commands from the brain and subsequently leading to atrophy. And it has been shown that there is significant neurotoxicity in these nerves that lead to this deterioration. The average onset is of about 50 years, leading to subsequent death and severe clinical disability in the subsequent years. Prevalence is about 5 to 100,000, leading to 60 to 65,000 patients worldwide. And it's been shown in recent publications very clearly that oxidative stress and accumulation of TDP-43 aggregates play an important role in the pathology of ALS. VectorY aims to address these pathologies through three pillars: using vectorized antibody fragments, using vectorized technologies and improved tissue and cell-specific engineered capsids to do so, and taking a strong effort in developing proprietary manufacturing, both in terms of the process and also in terms of the infrastructure, with the aim to develop large-scale, low-cost manufacturing platforms. Our concept is explained here on this cartoon. So on the left-hand side, you see vectorized antibodies. We tend to use the short single-chain FEs delivered by an AAV directly into the target tissue, into the nucleus, and then expression into the cytoplasm, where these fragments bind to the aggregates, the brown bulbs again. And these will then further lead to degradation into this in cytoplasm. This process can be accelerated, as is depicted here in the form of this little key, to be directed directly to the proteasome. And that concept is we call PROTACs, in analogy to PROTAC, to accelerate degradation of the aggregates. At the same time, we know that these aggregates tend to spread in a prion-like fashion through the extracellular vesicles. And we anticipate that also our PROTACs, vectorized antibodies with the antibody associated, will travel into other tissues rather than the direct site of administration or the direct tissue affected. And so we will anticipate to see a broader and distributed therapeutic effect. We are using a vectorized gene therapy approach to address imbalances in the proteostatic system in three areas: reduction of oxidative stress, blockage of the formation of toxic protein aggregation, and also reduction of the aggregate protein load through accelerated degradation. Oxidative stress leads to the formation of oxidized phospholipids and subsequent motor neuron axonal damage in ALS. The oxidized phospholipids get recognized by the immune system as foreign, and therefore these epitopes can be recognized by an antibody like EO6 that interferes with the oxidized phospholipids mediated inflammation and tissue damage, both in the periphery as well as in the CNS. Our AAV vectorized EO6 antibody overcomes the issue of insufficient CNS targeting by direct linking to the specific target tissue. Preclinical models from other AAV-based platforms have demonstrated that it's possible to achieve attractive brain distribution and expression of EO6 under such an approach. That is more depicted here. Here you see that oxidized phospholipids induce indeed toxicity in human motor neuron cells and muscle cells. On the top, you see the iPSC-derived motor neurons, and at the bottom, skeletal muscle cells. On the left, two panels in a non-treated form, and on the right-hand side, after exposure of oxidized phospholipids, showing on the top the shortened neurons, and on the lower panel, the thinner fibrils. That is quantified on the right-hand side, and you see average outgrowth per cell in a dose-dependent manner being reduced upon exposure to oxidized phospholipids. And at the same time, at the bottom, you see muscle cell viability being reduced upon such exposure, also in a dose-dependent manner. Furthermore, oxidized phospholipids induce axonal toxicity and tend to concentrate there, specifically in ALS and frontotemporal dementia, as is shown here. On the left-hand side, left panel, and the left side, you see the tissue samples being stained with the oxidized phospholipid antibody for oxidized phospholipid antibody presence. And on the right-hand side, you see these stains actually concentrating in the axons in both ALS and frontotemporal dementia, and not so much in the Parkinson's disease example. On the right-hand side, you see the microscopic phenomenon being illustrated with EO6 and amyloid precursor protein, a proxy for toxicity, concentrating in the axons, both in ALS as in FTD. So clearly showing the pathology concentrating in the axons and related to oxidized phospholipids. So our approach is trying to reduce this oxidized phospholipids and therefore aim to achieve a therapeutic effect. Alternatively, we tend and aim to produce blockage of the formation of toxic protein aggregation as well. And one example here is TDP-43, which is a ubiquitous protein intranuclearly, but when it goes and leaks into the cytoplasm, it can actually resort to toxic effects, specifically the aggregates. And it has been shown in their AAV9 single chain FE that the cytoplasmic TDP-43 aggregates can be reduced in an effective manner, as is shown in the left-hand panel. On the right-hand side, in a preclinical model of ALS mice, it is shown that the cognitive deficits can be improved. And what you see here on the left-hand side, the white is a control non-ALS animal. The gray is an ALS animal, both exploring both familiar and novel objects, so no additional challenge through novel objects, i.e. curiosity. And in the treated pink dots, you see a significant improvement based on this AAV9 platform. And so it is shown that not only can you reduce the TDP-43 aggregates, but also the cognitive deficits. And we will use an improved version of this going into our development program. At this point, VectorY has already assembled a number of antibody fragments that recognize specific TDP-43 epitopes across the genome, and we're testing which one would be most effective. We're using multiple VectorY antibodies against the TDP-43 to reduce cytoplasmic TDP-43 aggregation, as shown before. And it's shown here in these pictures that compared to control 1 and 2, our versions V1 to V13 can actually reduce protein aggregation in vitro already quite effectively from the optic assessment. Secondly, VectorY aims to develop under a rational engineered approach, AAV-based tissue and cell-specific capsids that have a broader application than just in the neurodegenerative space. And here is an example of doing that for skeletal muscle cells, where we show in a dose-dependent manner that the virus transduction efficacy can be significantly improved for myoblasts as well as for myotubes. And we're continuing to work today to further improve on that, showing that our technologies can be applied across a wider range of potential indications. And finally, we're focusing heavily on the development of proprietary manufacturing capability, both in terms of the process, as is depicted here schematically, where we're doing a very disciplined approach on redesigning AAV-based production process for baculovirus insect cell-based platform here for the USP part of the process, integrating the latest insights into those processes, and including also online monitoring and in-process analytics to improve and guide the process. We aim to do that in a pilot plant setting, up to 50-liter disposable bioreactor systems. Now we're going through a similar approach under the downstream processing part of the process. Furthermore, we aim to develop a early clinical and early commercial manufacturing capability, going from left to right, as I earlier described. We are setting up and have set up our process development laboratories in Amsterdam Science Park, where we started our operations in October 2020. And we are now moving forward to finalize the work there. We are also now in the process of closing an agreement with a large CDMO to establish a very large gene therapy facility in which VectorY will have access to a dedicated part of that GMP facility, where we could then produce up to 2,000-liter disposable bioreactor processes for clinical and early commercial manufacturing. So in terms of the programs, ALS as a proof of concept disease, we will approach through oxidized phospholipids as well as TDP-43 aggregate program in O1 and O2 program. Our rationale for that has been that there's important validation that the neutralization of oxidized phospholipids can result in therapeutic benefit in several diseases, and also in a recent mouse brain model. Ameliorated oxidized phospholipid toxicity can be observed. And we believe that lowering of the oxidized phospholipids may lead and will lead to a therapeutic effect. TDP-43 aggregates, the rationale is delivered by the fact that these are present ubiquitously in the ALS patient population, and therefore also reducing the cytoplasmic TDP-43 aggregates will most likely have a positive effect. As is shown in the data that I've seen, it seems from earlier preclinical models, that a cognitive effect, positive effect, can also be expected. So our pipeline is developing as follows. So O1 and O2 have been described, both oxidized phospholipids and TDP-43, where we aim to be in the clinic in 2023. We have a partnership established for the oxidized phospholipids with a world leader in the antibody technology associated with that, and we're closing a second partnership on TDP-43 with another renowned specialty group who knows this space very well. And then at the moment, we're going through an assessment of our further capabilities and target disease assessment, where we will aim to extend our pipeline to a third and fourth program. As you know, all of this is possible since October 2020 through a very strong management team with a deep experience from several companies for many, many years, like Unicure, Janssen, and other biopharmaceutical leaders in order to accelerate and provide stability to our program. So in short, VectorY is extremely well positioned to develop innovative therapies for these disorders, neurodegenerative and neuromuscular, focusing largely on the theme of proteinopathies, using vectorized antibodies, improved capsids, and proprietary manufacturing, with a strong management team and already a very strong capital base. Thank you very much for your attention.